All right, everybody. This is going to be the last of the review videos for exam 3. So this is going to be chapter 16, which is mostly going to be about DNA replication. But before we get into the process of DNA replication, to understand this process and understand this chapter, it's really, really vital that you have a pretty good understanding of the structure of DNA. So if you remember, DNA, which is deoxyribonucleic acid, is comprised of monomers that we call nucleotides. That says monomers, I spelled it horribly, but monomers are nucleotides. Nucleotides. And hopefully you kind of remember from way back in the first couple chapters, the structure of a nucleotide. So we have a nitrogenous base, which is either adenine, guanine, cytosine, or thymine. A lot of times those will just be shortened to A, G, C, and T. So we have our nitrogenous base, which is attached to a 5-carbon sugar. And that 5-carbon sugar has, this is going to be really important, has a hydroxyl group, which is that OH minus group. It has a hydroxyl group on its 3' sugar. And so that side of the nucleotide, so this side down here, is going to be referred to as the 3' end. And then on its 5' sugar up here, it has a phosphate group. And the end of the nucleotide that has this phosphate group on this 5 sugar is going to be called the 5' end. And that's going to be really, really important that you are able to orient that properly. And we will get into why in this part down here. So the next thing that I want you to remember is that these nucleotides link together, right? So there will be a whole bunch of these in tandem, and we'll get into the bonds down here as well. But I just want you to remember that it forms these 2 strands. So this is 2 strands of DNA. And when we have a strand, because you remember the shape of DNA as we kind of think of it is this double helix shape. And this double helix shape is really comprised of 2 complementary strands of DNA. And that means 2 strands of DNA that complement each other, that have certain nitrogenous bases pairing with very specific other nitrogenous bases. And you may remember that A's or adenine always bonds to T, which is thymine, and cytosine always binds guanine. So we have these 2 complementary strands, but the other, and remember we have this line right here is sort of the phosphate and sugar backbone. So that's this part of the nucleotide. And then these the rungs of the ladder sort of are the nitrogenous bases. I also really want you to remember that these strands are anti-parallel complementary strands. And that is going to be really important for replication too. So we have this strand, which is oriented with its 5' end up here, which means off of this, like on this very last one, we would have a free phosphate. And then its 3' end is down here, and its 3' end would have a free hydroxyl group. But this complementary strand is actually reversed. The 3' end is at the top here, and the 5' end, which would have the phosphate, is on the bottom. And this is important because DNA, when we are building DNA, when we're linking these nucleotides together, we can only link them in the 5' to 3' direction. I'm going to say that again. When we are building DNA strands, when we are adding nucleotides together, we can only add nucleotides. A new nucleotide can only be added to the 3' end of the molecule. And molecularly, when we think about that, that means that in order to join two nucleotides, we are going to be adding a nucleotide to this OH group, to this 3' OH group. So if I wanted to add a nucleotide, if I wanted to bind this nucleotide to another nucleotide, I would have to attach that new nucleotide down here on the 3' end. So remember, the direction of synthesis is always 5' to 3'. So down here, we're going to get into the bonds of these DNA molecules and how these nucleotides link to each other, because there's 2 linkage points. So there's going to be the linkage that forms the sort of sides of the ladder, which are these phosphate sugar backbones. And then there are also going to be bonds between these nitrogenous bases, between the rungs of the ladder. So if we look here, I have two nucleotides on each side here. And I've labeled, so on this side, we're oriented with the 3' end here and the 5' end here. And remember, the 5' end has this free-floating phosphate, and the 3' end of the strand has this free-floating hydroxyl. And so if this side is 5' on top, 3' on bottom, the complementary strand has to be anti-parallel. So it's going to have 3' on top, which means it has this free hydroxyl group, and 5' on the bottom, which means it has a free-floating phosphate. So I have drawn there 2 types of bonds that matter when we're talking about DNA, and that is going to be the bond between a phosphate group, a 5' phosphate group, and a 3' hydroxyl group. And that's what's going to bind the sort of the backbone of the ladder, these sides of the ladder. And those are phosphodiester bonds, which are covalent. So remember that phosphodiester bonds link sort of the backbone, link this phosphate to this 3' OH group on the sugar. And you may remember I said we can only synthesize from the 5' end to the 3' end. So when we look at these 2 nucleotides, we know that I added this bottom nucleotide, I added it on to this. It's not the other way around. You need to think of this as I attached this bottom nucleotide to this 3' OH group. I could not attach a nucleotide up here. I would have to go this way on this strand. The direction of synthesis would be going down relative to my paper. So if I wanted to keep adding nucleotides, I would be adding them down here. I would add something onto this OH group, and then I would add something onto the next OH group. I would never be able to add a nucleotide to this phosphate. So if this direction of synthesis was down this way, that means this anti-parallel strand is going to do synthesis the opposite way because it's anti-parallel, and we can only go in the 5' direction to the 3' direction. So here we have a nucleotide. This is thymine. I just wrote out the nitrogenous bases, and we'll get into those bases in a second here. So we have another phosphate group that I have added onto this OH group, and that is another phosphodiester bond right here. So that phosphodiester bond is linking this nucleotide to this nucleotide. And remember, if I wanted to keep adding nucleotides to this strand, I would have to add them onto an OH group. So I would keep adding them in this direction, up. So that's the that's sort of the phosphate sugar backbone. So now we're going to talk about this middle portion, and we're going to talk about our nitrogenous bases. So you can see structurally we have all 4 bases. I have guanine, which binds cytosine, and I have adenine, which bonds to thymine. And there are a couple classifications that I want you to think about. So this adenine and guanine here, so adenine on the bottom and then guanine right here, you can see that they have 2 ring structures. So adenine and guanine have double rings. You can see we have sort of this 5 carbon structure and then a 6 carbon structure added onto it. So guanine and adenine are called purines. That's just what they are classified on, and they have 2 rings. So purines are right here. So we have purines in adenine and guanine, and they have these 2 ring structures. On the other side, we have thymine and cytosine. And thymine and cytosine only have one ring. They only have this 1 6-carbon structure. So cytosine and thymine are the pyrimidines, and they have 1-ring structures. So I always think of it as thymine and cytosine both have Y's in them, and pyrimidines have a Y. So when you're trying to think, oh, which ones are pyrimidines and which ones are purines, pyrimidines are going to be the ones with Y's, cytosine and thymine. And it's really important to understand that this 2-ring structure and this 1-ring structure, because you'll notice that purines, the adenine and the guanine, bind to the pyrimidines. And the reason that they do this is it's spatial organization. If we tried to bind a guanine to an adenine, this 2-ring structure takes up more lateral space than a 1-ring structure. So there would not be enough room between the sides of my ladder, between these phosphate sugar backbones, to have that much size, to have 2-ring structures. So the solution is these 2-ring structures bind to a 1-ring structure, because that's just sort of just right. You can think of it like Goldilocks and, the 3 bears. Adenine and guanine, we don't have enough space. If we tried to bind cytosine to thymine, so if we tried to bind a 1-ring structure to another 1-ring structure, we would have too much space in between the rungs of our ladder. So instead of the rungs of our ladder fitting sort of nicely close together like that, they would be like this, and they wouldn't be able to attach to each other super well because of all this empty space. So really what we look like here is we actually have sort of one that extends a little further and then one that's a little shorter. And this would be your purine and this would be your pyrimidine. If we have 2 of these purines, then there's too much space in between them. There's not enough space to house both of them. It would have to sort of push this backbone out, and we don't want that. So that's purines versus pyrimidines. So now we're going to talk about the specific bonds. It's important to know that adenine always binds to thymine. Adenine will never bind to cytosine. So it's always A to T, and it's always G to C, and vice versa. Cytosine would always pair up with guanine, and thymine would always pair up with adenine. And something that I want you to remember about these 2 bonds, the adenine-thymine bond and this guanine-cytosine bond, is that adenine-thymine bonds are held together by 2 hydrogen bonds. So we have 1 hydrogen bond here, and we have 1 hydrogen bond here, for a total of 2 hydrogen bonds. Shouldn't have put a plus there. But the cytosine-guanine bond has 3 hydrogen bonds. Okay, so cytosine-guanine has 3 hydrogen bonds. And I wrote that down here as well. So AT has 2 hydrogen bonds and CG has 3 hydrogen bonds. So that means that this CG bond is actually stronger than the AT bond. So if you ever get a question that asks anything about the strength of a bond, know that the CG bond is going to be stronger than the AT bond, which means it's easier for me to pull AT apart as opposed to CG, because I have one more bond that I have to pull apart to separate a CG bond, a cytosine-guanine bond. And so that's the second type of bond. So remember, we have phosphodiester bonds on the backbone of the DNA molecules. And then joining the nitrogenous bases are hydrogen bonds. Okay, and we already sort of covered this. Remember that these DNA strands are anti-parallel. So we have 5' 3' over here, but then it switches. We have 3' on top here, 5' over here, which also means the direction of synthesis, since DNA can only be synthesized 5' to 3', switches. So we have synthesis going down on this side, and synthesis would travel up on this side if I wanted to add nucleotides. So now that we've gone over sort of the structure and the molecular DNA stuff, we can get into replication. So replication has 3 steps. It's going to be initiation, elongation, and termination. So the first thing that happens when we have DNA replication is we have our strand of DNA here that I want to replicate. And remember that they are anti-parallel. So I have 5', 3' here, so 5' on the left, 3' on the right. But then this complementary strand is 3', 5'. So when we start replication, so you also see up here, I have written out sort of the key enzymes, and we'll talk about them as we go. So the first enzyme that's going to act in initiation is helicase, or I think it might be DNA helicase. And DNA helicase, I want you to think, is the unzipper. So what's going to happen here is DNA helicase is going to insert at one point, and we are going to call that point the origin of replication. And here, these 2 enzymes right here and here are going to be our DNA helicases. So that's helicase and that's helicase. And what these helicases are doing is they are going to go in each direction. So we have one that's going to go this way and one that's going to go this way. And it's going to start breaking apart the hydrogen bonds between nitrogenous bases. So helicase is unzipper, breaks nitrogenous base hydrogen bonds. And when we do that, it's going to sort of cause these DNA strands to sort of float away from each other and open up a little bit. And this is going to form what's called the replication bubble. And then we're also going to have our replication forks on each side. So we have a fork, because remember, it's kind of looks like this. So these would be the forks right here and right here. So we now have this replication bubble and we have these DNA strands that are sort of opening up. But what we don't want to happen is we don't want these hydrogen bonds, so I don't want this base and this base to pair up again. I want to keep them separated. And so I'm going to do that by using single-stranded binding proteins. So single-stranded binding proteins stop bases from re-pairing. Not repairing, re-pairing. So it stops these H bonds from forming. And these single-stranded binding proteins are just kind of going to attach on to nucleotides like this. And then we'll remove them when we want to add new DNA. So those are single-stranded binding proteins. All right, so the next enzyme that I want to talk about, and I wrote them out of order, unfortunately, a little bit, and sort of the last enzyme for initiation is going to be topoisomerase. So topoisomerase is going to stop DNA from bunching up. And what I mean by that is you can kind of think of this as if you took, let me see if I have something to demonstrate this. All right, sorry about that. So to demonstrate this, I have brought my shoe. So there's a problem with this whole helicase opening up the replication bundle, or the replication bubble. So you can imagine that the DNA is sort of, let me sort of go this way actually. DNA is sort of wound around each other. We have our 2 complementary strands, which is going to be my 2 shoelaces. And it's sort of wrapped around each other a whole bunch of times. So it's kind of like that. So what helicase is going to do is it's going to come in at any point and it's going to start unzipping the DNA, or it's going to start unwinding my shoelaces. And it's going to try to do that pretty fast. But you can see if I have this strand and I just start trying, hold on, let me get it nice and really, really wound up. So helicase, when it tries to unzip the DNA, what can happen is when I pull, you can see I get to a point that it sort of gets stuck. Like I can't pull this apart anymore because I have this huge bunch up that's happened. And so no matter how much I pull, that's just going to tighten it. So topoisomerase is sort of going to act against that. And this would be topoisomerase. It's going to be acting sort of further up the strand, so further up. And it's just going to kind of like pull the DNA a little bit so that I can keep unwinding. I can't super do it with one hand. But if we have something sort of relieving the pressure that happens when I do that, then it can start to unwind a little better, and it can do its thing better. So that's what topoisomerase's job is. Topoisomerase is going to be over here, and then there would be one on each side. So we'll call this the topoisomerase. So topoisomerase is sort of stopping the DNA from getting stuck during this unzipping process as this helicase moves and continues to unzip. Topoisomerase is acting as a reliever. And that's it for initiation. So initiation is really just the process of opening up the DNA, creating the replication bubble, and getting ready for the next phase, which is going to be elongation. So elongation, we're going to start up here. And what I've done here is we're only looking at that top template strand of DNA right now. So just understand that there's going to be, because we're replicating this DNA, so there's going to be one strand that's going to be built off of this top strand, and that's the one that I'm going to look at. But it's also going to be happening simultaneously with this bottom strand, and I'll illustrate that sort of near the end of the video. But just for simplicity's sake, we're only going to look at this top strand now. So elongation. Elongation is the actual adding of nucleotides to create a new strand. So remember we had our origin right here, and then we had the DNA sort of unzipping in each direction and creating this bubble area. So if I want to start making a new strand, I'm going to build off of this existing template strand. So this is the template strand. And remember though, I can only build in the 5' to 3' direction. And remember that DNA is anti-parallel. So I'm going to create a strand. So if this strand is 5' on the left, 3' on the right, the strand that I'm creating to sort of bind to it will be 3' on the left and 5' on the right. So you remember that we had this origin point and we unzipped in both directions. That creates a little problem, because when I'm making my new strand and I'm going 5' to 3', so I can only build my new strand going from right to left, that's going to be easy when I'm going left of the origin. So when I'm going left of the origin, I can just sort of continually make this strand from 5' to 3'. But if I'm going to the right of the origin, so if I'm going this way doing my synthesis, I can't continuously build that way, because that would be building in the 3' to 5' direction. And like I tried to hammer it in, you can't do that. We cannot build DNA 3' to 5'. So what I have to do is sort of build it in little segments as the DNA unzips. So as this DNA keeps unzipping, I can sort of build backwards toward the origin. And that's going to produce what's called a lagging strand. And we'll get into why it's called a lagging strand in a little bit. But just know that lagging strand is built in fragments. And so you can see that we have a leading strand that is built away from the origin of replication. So this leading strand I'm going to build in this direction, but the lagging strand I have to build in fragments toward the origin of replication. So we're going to start with the leading strand because it's a lot easier. So the first step, so we have our origin here and we have our template strand. So it's really only a couple steps for the leading strand. It's very simple. So the first thing that's going to happen is we're going to have, this should be DNA primase is going to lay down an RNA primer. And what an RNA primer does is it gives a start point for this next enzyme to start adding nucleotides. So we're going to attach this RNA primer right here, right where we want to start replication, right at the origin for the leading strand. And that's going to be done by DNA primase. So the next phase is we're going to have another enzyme, DNA polymerase 3. So DNA polymerase 3 is going to attach to that primer, and it's going to start moving from the 5' to 3' direction and adding free-floating nucleotides. So DNA polymerase is going to go through here, and let's say that this is an A, this is a T, this is a C, this is a G, this is an A. DNA polymerase is going to start at this primer, and it's going to go from there, and it's going to go 5' to 3', so it's going to go, okay, there's an A right here, let me grab a free-floating T, a free-floating thymine nucleotide, and let me just attach it right here. Boom. And when it does that, it's going to create the phosphodiester bond. So it's going to attach to a hydroxyl group. And then it's going to these will spontaneously bond, this T and this A. And it's also worth noting at this point, the single-stranded binding protein that was keeping those DNAs apart is removed. So DNA polymerase is going to remove that single-stranded binding protein, and then it's going to attach the complementary base. And it's just going to keep going through that. It's going to go, okay, there's a T here, let me grab an A and put it here. There's a C here, let me grab a G. There's a G here, let me grab a C. There's another A here, let me grab a T. And it's just going to work in that 5' to 3' direction, synthesizing new DNA and attaching it. And remember, it's going to be attaching, so this guanine is going to be added to the 3' hydroxyl of this adenine. And then this cytosine is going to be added to the 3' hydroxyl of this guanine. And it's just going to keep adding things to 3' hydroxyls until something tells it to stop, which oftentimes will be another primer. It might be another enzyme, like maybe it runs into another replication bubble and that'll stop it. But that's DNA polymerase 3. And the last thing that we're going to do is we have this primer here, and we didn't synthesize anything for these 3 nucleotides because the primer had to be there. So DNA polymerase 1 is going to remove that primer. It's going to go in and it's going to take this primer off. It's going to grab this, remove the primer, and it's going to add nucleotides to these missing parts. So if this is an A, a T, and a G, then DNA polymerase is going to get this primer out of here, and then it's going to go, okay, here's a thymine for your adenine, here's an adenine for your thymine, here's a cytosine for your guanine. And it's going to create that little fragment. And then the last enzyme is going to be DNA ligase. And DNA ligase is the rezipper, or sorry, not the rezipper. I think of DNA ligase as as just the binder, is how I would say it. Because now we have these 3 nucleotides. We have this cytosine, adenine, thymine, and I believe this was a thymine right here. But this cytosine is not connected to this thymine. We're missing this linkage. We're missing this phosphodiester bond. And remember, we can't just spontaneously attach that because we would have to be adding something to this 3' hydroxyl. So to solve that, we have to use a special enzyme, DNA ligase. DNA ligase is going to grab these 2 strands. It's going to grab this thymine and the cytosine, and it's going to fit them together and complete the strand. So now we have a cohesive strand that is complementary and anti-parallel to our template. So that's the leading strand. Now we have the lagging strand synthesis, which is going to be a little more difficult. And you remember, this is the one I said is going to be built in fragments. So we have our point of origin here, and we have our 5' end of the template and our 3' end of the template. So when I'm building an anti-parallel strand, I have to build from 5' to 3'. But that's not the direction that this DNA is unzipping. This DNA is unzipping and wants me to replicate it in this direction, from left to right. But I have to synthesize from right to left. So the way we do this is in little fragments, which are called Okazaki fragments. And we'll get into that. I write that down later too, but these are Okazaki fragments, named after the guy who discovered them, I'm pretty sure. So the first step to build the lagging strand is going to be DNA primase. It's the same enzyme, but instead of putting one primer at the origin, as this DNA is unzipping, it's going to keep putting primers down every so often. Every time it sees a certain DNA sequence, it will lay down a primer. So the more this DNA unzips, as we keep pulling this DNA apart as it travels down here, we'll start attaching more and more primers. So now that we have those primers as it's unzipping, the next thing is going to be that DNA polymerase 3 again. So same enzyme that was building the leading strand, we have our primers, is going to attach here. It's going to attach to this primer. Another one might attach to this primer, and it'll attach to this primer. And it will start synthesizing in the 5' to 3' direction of our new strand. So it's going to attach to this primer, and it's going to go, okay, maybe this is an A and maybe this is a T. And it's going to go, okay, here's a thymine, here's an adenine, boom. And then sequentially, this next primer will be placed down as this unzips, and another DNA polymerase 3 will bind to it and keep synthesizing DNA in the 5' to 3' direction. And then maybe it unzips a little more, and we have another primer that gets laid down. And once that primer is laid down, we can go DNA polymerase 3 and start synthesizing 5' to 3'. So the last step, and this is sort of the termination step, and I should have said that back here as well. This DNA ligase binding, this last step, is termination. We're ending the synthesis. And so now the termination phase for the lagging strand looks a little bit different. So we had all these primers and these Okazaki fragments. So now DNA polymerase 1 is going to go through, remove the primers. So DNA polymerase 1 is going to take this primer away, and it's going to add the correct bases. But you may remember when we had this happen with the leading strand, it left this gap that needed to be connected. It left this gap in the phosphate sugar phosphate backbone that DNA ligase had to come seal. So that's going to be the exact same thing here, where DNA ligase is going to come in. So this is ligase, is going to join these backbones. And then another ligase will join these backbones, and then ligase will join these ones, and so on and so on. So that's lagging strand synthesis. And now I want to get into sort of why it's a lagging strand. So here is the full replication bubble. So we have our template strand on the top here, and we have the template strand on the bottom here. And these are the two strands that are being pulled apart at the start, right? So on each side of the origin, we're going to have one leading strand being synthesized and one lagging strand being synthesized. And I want you to imagine this exact point in time right here. So we're going to just go like this. So at this point in time, nothing has been unzipped here. I want you to look at the stage of replication of each strand on this side. So our leading strand has gotten to this nucleotide right here. It hasn't gotten further yet because we haven't unzipped that yet, we're saying. But on the bottom, this lagging strand, this nucleotide, has not been synthesized yet. This nucleotide has not been added to a fragment because we have not unzipped this DNA enough to get to this primer. So this primer, at this point in time, this primer has not been placed, so this fragment can't be synthesized. So this isn't happening at this exact point in time. And so we have a leading strand that has gotten to here, but this lagging strand has only been able to get to here because it doesn't have the next primer. And so that's why it's a lagging strand. So the next concept, so that's it for replication, but we're gonna sort of talk about a few structural things now. So you have on the ends of your DNA things called telomeres, and telomeres are these protective caps that are just repeating DNA sequences that don't code for anything. And the reason that we have these is because with each replication, the DNA molecule is going to shorten by 1 primer length. And the reason that that's going to happen is because whenever a DNA polymerase wants to add DNA, it has to see a 3' hydroxyl group to attach a new nucleotide to. So if you imagine down here, if I just have the end of my DNA, so we have 5' to 3', and then we have 5' to 3' on our new strand that we're synthesizing. This primer right here, when this primer gets removed, there is nothing here for DNA polymerase to attach a new nucleotide to. It doesn't have a 3' hydroxyl group to attach to, so it cannot put new nucleotides right here. And that is why your telomeres would shorten. However, there is an enzyme called telomerase. Some people say telomeres, some people say telomeres. I say telomeres, so I would call this telomerase. Call it whatever you want. Just know, all I want you to know is that telomerase can solve this issue of shortening telomeres. And it does that by sort of including an extra primer, if you can think about it like that. It includes a little bit of extra RNA, and that RNA will have a hydroxyl group that DNA polymerase can attach to. So that's all I want you to know about telomeres and telomeres. Okay, we're almost there. Last page. So these are mutations. So mutations occur when you have bad DNA replication, when something in DNA replication does not go properly. So I want you to imagine we have a template strand that reads A, C, C, T, G, A, G. You should be able to write out complement strands. So if a test question asked you, like, what's the complement strand to this template, you would go, okay, well, first of all, the first thing I want to check is anti-parallel. So if this is 5' and this is 3', then that means that this one is 5' and this one is 3'. And then you have to read and just do the base pairing. So an adenine would bind a thymine, a cytosine would bind a guanine, and you just keep going. So the correct replication would be T, G, G, A, C, T, C. And I just want to do a quick aside here. On tests, a lot of times they will ask you a question that says, what is the complement to this DNA sequence? And maybe it just goes something like that. Be really, really careful when you are answering, because a lot of time, and the thing I want you to really keep track of if you see a question like this is your 5' end and your 3' end. Because let's say that maybe I could write this the way I would intuitively write this to find the complement strand would be I would put 3' here, and I would go, okay, A would be T, T would bind A, G would need a C, G would need a C, T would need an A would need a T, and then this would be the 5' end. So we have our anti-parallel strands. On multiple choice tests, a lot of times professors will write strands in the 5' to 3' direction. So the way the answer might look on your test would actually be like this. I picked a really bad strand for this because it reads the same forward and backwards. I'm going to change this to a C and this to a G. So sorry. So they might write it 5' to 3' because most of the time people write DNA sequences in the 5' to 3' direction. So if it gives you the template strand that's 5' ATGGTC, I would write out the complement strand as 3' TACCAG, but the way the answer might look would be would essentially be reading what I just wrote backwards and going 5' GACCAT, which would look like this. Be really, really careful about your ends, because there will be questions, and I think on Dr. Reddy's exams last year when I was in the class, she wrote one answer like this and then wrote another answer like this, which would be incorrect, because we would need to flip these. So just be careful. Anyway, back to what we were doing. So our correct replication would produce a strand that reads TGGACTC. So there are 3 types of mutations that I kind of want you to think about. And they're pretty intuitive, but we'll go over them real quick. There's substitution, insertion, and deletion. Substitution means I mispair something, basically. So instead of this C making a G, this C actually made an A. So now it's TAGACTC, and that's a substitution. An insertion means I just add an extra nucleotide. So maybe a cytosine gets inserted between this guanine and this adenine. So that produces this strand. That's an insertion. And a deletion is I just remove a nucleotide. So instead of TGGA, I might end up with TGA, and I'm missing this G. That would be a deletion. So these are the 3 main types of mutations, and then we have chromosomal mutations as well. So now we're imagining the whole chromosome. So a chromosomal deletion would mean that, so we have these segments, 1, 2, 3, 4, 5, and 6. So a deletion would mean this chromosome that read 1, 2, 3, 4, 5, 6. We miss this 3rd segment here. We just go 1, 2, 4, 5, 6. An inversion means we take a portion of this chromosome and we turn it on its head. So instead of my pen going this way, we take it out, we flip it, we put it back in. And so that would be like this 1, 2, 3, 4, 5, 6 going to 1, 4, 3, 2, 5, 6. So this 2 through 4 segment got inverted. A duplication just means we duplicate one segment. So 1, 2, 3, 4, 5, 6 goes to 1, 2, 1, 2, 3, 4, 5, 6. So we had a duplication of this 1 and 2 segment. And the last one is translocation, which means we move one segment. We move a segment from one chromosome to a non-homologous chromosome. Non-homologous. If the definition did not say non-homologous, then we would just be talking about crossing over. So it is non-homologous. So translocation, so let's say we have your 2nd chromosome reads 1, 2, 3, 4, 5, and your 15th chromosome reads 6, 7, 8, 9, 10. Translocation means these non-homologs swap portions of DNA. So maybe it swaps this 6, 7, 8 for this 1, 2, 3. And now on this chromosome, we have 6, 7, 8, 4, 5. And on this 15th chromosome that we had 1, 2, 3, 9, 10. I also want to talk about mutations. This will be just verbal, very briefly. Not all mutations are bad. Some mutations are actually good. They can either have a positive effect, a negative effect, or no effect. And if it has no effect, that's called a silent mutation. And I think that is it. So this was the last one for your third exam. So good luck on those exams and have a good night.